get started. So basically, you should have had an online workshop around this great metabolic race. Not sure if you actually have had it. Last time it was actually optional for us. So if you haven't done it, don't worry, I'm going to explain what it is so you actually understand what's going on. All you have to know is that you're going to pretend that this is a race, okay? We're going to pretend it's a 10-kilometer race and someone's actually running, okay? So we have this diagram here, which would have or should have been given to you during the workshop, okay? Oh, sorry, switched too fast. Yeah, so basically here we have in minutes the progression of the race at the bottom, and here we have the percentage of energy supplied, okay? So from 0 to 100%. Now we have two lines here. We have a line for carbohydrate use and a line for fat use. Okay, and basically this diagram just details where we source our energy from, whether we source it from carbohydrates or from fats. Oh, okay, I see someone said that you actually have had the workshop, which is great. So today I'm not going to go into all the detail about all the questions that you've gotten. When we did it, it was a bit too complicated and it's too much that you need to know. You just need to understand sort of the general principles behind it. So that's what I'm going to be covering today. So at the beginning, okay, so pretend you've just rocked up to the race and you haven't started running yet, okay? You're just relaxing beforehand. So we know that in normal metabolism at rest, we mainly use fat, okay, or lipids as our primary energy source, okay? And remember particularly that the heart and skeletal muscles use these lipids, okay? And that's where most of it is coming from. However, we can't forget that we also use carbohydrates, okay? So remembering that our brain and our red blood cells still always pretty much need to use glucose, okay, or preferably glucose. So this is why there's still a little bit of carbohydrates at this 0 minutes at rest, okay? Now, pretend that we actually start running the race, okay? And you're using suddenly a lot of energy, okay? So you should probably remember that we undergo anaerobic respiration, okay? So that's in the absence of oxygen, and we're converting the glucose and we're producing lactate, okay? Now, the reason why we start off with that is because it's a very sudden demand on the muscles and we can't get enough oxygen that quickly to our muscles. And also it's a very quick way of producing energy, okay? So even though you produce only 2 ATP, it's a very quick reaction and it's an immediate source of energy. So that's why we start off with anaerobic respiration. Now, after here, you can see that our carbohydrate use is steadily increasing, okay? Now, we can't undergo anaerobic respiration forever, okay? Because firstly, you get a buildup of lactic acidosis, so you don't want that, that's not good for you. And then your muscles can't indefinitely not use oxygen, okay? Because they still need an oxygen supply later on. So that's why we convert to aerobic respiration. And so we get this glucose mainly from either our diet, okay, so there still might be some leftover glucose coming from the energy bar you just had before the race, or after a while, that will be depleted. So then we turn to our glycogen stores, mainly in our muscles, okay? And that's why you can see our carbohydrate use is going up. We're undergoing aerobic respiration. We're taking that glycogen and converting it to glucose, okay? Oh, sorry, jumping forward, that's not good. So we keep doing that, and then we reach this sort of peak, okay? So this is literally in the first couple minutes, 5 minutes, okay? And then you can see we start using less glucose. Now the reason for that is because you can think about this. If we've used a lot of glycogen, we're going to have less glycogen left in our muscles. We're using up those stores, okay? So we start relying less on these glycogen stores, so we get less carbohydrate source, okay? Now, you still need a lot of energy, okay? So if you're getting less glucose, you need to get that energy from somewhere else. So that's why we turn to lipid sources, okay? So remember, at the beginning, we're not really using lipids anymore, okay? We have an increase of use in our muscles, so we really need that glucose and we're not relying on that. But after a while, to sort of supplement this depletion in glycogen, we start using lipids more and more and more, okay? At 30 minutes, okay, you can see it's at 50%, you're using the same amount of carbs and fats as your energy source, okay? And so this is sort of an important crossroads to remember. So if you want to remember 30 minutes is where you end up using around about the same amount of carbs and fats. Okay. Now, after 30 minutes, you notice that we start primarily relying on lipids. okay? And that's because, remember, you have quite a substantial store of fats in your body, okay, from adipose tissue. So you can almost imagine fat burning. If you're doing aerobic exercise for more than 30 minutes, you're starting to use a lot of your fat stores, okay? And that's sort of the concept behind fat loss as well, because you're starting to use a lot of your fats from your adipose tissue to supply your muscles and everything else. But you're still always going to need carbohydrates, okay? Things like your red blood cells are always going to need glucose. So you never stop using carbs, okay? Even though we have an increase in lipids. Now, at the very end, it's not really in this diagram, it's a bit separate. Pretend you're running, okay? You're almost at the finish line, you're about 100 meters from there, and you really want to sprint from that finish line, okay? Now, we want this big burst of energy, okay? So what we use is something called creatine phosphate. So this should all be revision from the last presentation, but do remember, if you're sprinting and using a lot of energy, you're using creatine phosphate. Okay, there's only a limited amount of this creatine phosphate, okay, so it only lasts for a very short amount of time. Now remember that creatine phosphate converts ADP to ATP in one step, so it's a really efficient sort of way to get a lot of energy coming from this pathway, okay. So I hope that was a bit of a good overview, okay? You don't have to memorize this diagram, okay? If you understand the metabolism that we covered last week, you should be able to pretty easily know what happens, okay? So remember anaerobic respiration, and then going to aerobic, and then remembering that we primarily use lipids towards the end. So hopefully that was an okay sort of overview. If you have any more questions, particularly, I know sometimes it gets really confusing with that workshop. If you have any more specific questions related to the questions that they ask, feel free to message me. I'm more than happy to actually go over it individually if you'd like as well. So now I'm going to pass over to Hanyi, and she'll do a bit of a presentation on genetics. So, and this is just everything I said written down. Yep, so I'll stop sharing and hopefully she's got that. Yeah.
All right, thank you, Xenia, for giving us that massive overview. Yep, so I'm Hanyu, and I will just be taking you guys through genetics. There we go. Can you guys all see the screen? If someone can chuck it in the chat. Yep, okay, no worries. Awesome, so genetics, it's basically split into two subtopics, I guess. So the first part, we'll be talking about your population genetics. So that's going to include all about your SNPs and also your Hardy-Weinberg law as well. And in the second part, we'll be talking a bit about your chromosome disorders and also touching on pedigrees, which I believe you guys have an active learning on next week. So, there we go. Okay, so first on to population genetics. So first of all, introducing what is a monogenic disorder and what exactly is a SNP. So monogenic disorders, probably you can tell from the name, it's basically just a mutation in a single gene. So this can be dominant, recessive, sex-linked, or mitochondrial. So what is a SNP? A SNP is almost like a subset of your monogenic disorders. So as you can see, it's a common difference between our genomes. And in order for it to be characterized as a SNP, it has to occur in at least 1% of the population. So what that basically means is a SNP, for example, you can think of it as like an eye color or hair color. However, if we're talking about a specific disorder, such as like Turner syndrome or Klinefelter's, as we'll go into a bit more depth in a moment, those wouldn't be considered SNPs just because they don't occur that frequently in the population. And yep, so your SNPs, they're basically differences in your genes. So they can occur in your exons, introns, basically just means it can occur wherever on your DNA. And if they do occur in your exons, oh wait, yes, I think it's your exons, they'll basically actually affect your overall genotype versus introns, which will eventually be sliced out. So why are SNPs important? So there's essentially three main aspects, I guess. So first aspect, it will influence your disease susceptibility. So if you have a specific kind of gene, that's going to influence whether or not you are more likely to obtain a certain kind of illness, I guess. Second point, it's going to influence your response to medicine. So that's going to perhaps influence your reaction to certain kind of drugs. And the third aspect, it's going to open up avenues for personalized medicine. So if we know what kind of genes you have, that's going to allow us to recognize what kind of diseases you're more susceptible to. And then we might be able to essentially change your treatment so that we can make it more personalized to you as an individual. And that basically opens up many avenues for different kinds of treatments into the future. So this is essentially a slide just talking a bit about genetic terms. A lot of you guys would have learned this in biology, so I won't spend too much time on it, but just quickly going through it. So trait, essentially your characteristic of an organism. Phenotype, essentially talking about what you look like, your cellular appearance. Gene, something on your DNA. Locus is the location of that gene, or like a specific gene on your DNA. Allele is just a different version of your gene. Your genotype, examples are all shown over here. So you've got your capital A, lowercase a, and two lowercase a's. Homozygous just means you've got two same alleles at the same at a certain locus, and heterozygous just means you've got two different ones at a locus. So this is quite an important slide. So population genetics, it basically tells us about the prevalence of genetic disorders and also the frequency of carriers in a certain population. So it can also tell us a bit about essentially your likelihood of inheriting a certain kind of disease as well. So phenotype frequency and genotype frequency, it's pretty straightforward. Essentially, it's a proportion of people either with a specific phenotype or genotype in a given population. The three different types of genotypes, it's pretty much your capital A, a capital lowercase, and then your two lowercase ones. The way you work out the frequency, you essentially get the number of people in or with a particular phenotype or genotype, and you divide that by the total number of people. So we're looking either at percentages or proportions. When we talk about allele frequency, it gets a little bit more complicated, but still pretty much the same principle. So normally we use P to denote the frequency of all of your dominant alleles, and Q to denote all the frequencies of your recessive alleles. So because we're talking about proportions, all of them have to add up to 1. So I think this is back to like probability of methods. Everything has to add up to 100%. So proportions have to add up to 1. The way you work out your P, it's essentially looking at the number of alleles that you have in a given population. So because we're looking at the dominant allele A, there's two dominant alleles A for people with the genotype of capital A. So you go 2 times the number of people with the genotype of capital A. And then in individuals with a genotype of a capital A and a lowercase a, they've only got one dominant allele. So you only have to add that one number. And you essentially divide that by the total number of people, but you times that by 2, because every individual, they have 2 alleles for a certain kind of gene. And that's how you essentially get P. And the way you get Q, pretty much the same thing, except this time you're looking at your recessive alleles instead of your dominant. Okay, so now a bit on, okay, no worries. So Hardy-Weinberg Law. So what this essentially is, it's basically split into 2 parts. First of all, in the absence of migration, mutation, or selection, your allele frequencies do not change over time in a large randomly mating populations. So keep in mind, it has to be a large population and it has to be random. This will be important maybe for future questions, hint, hint. So second part, if your allele frequencies, they don't change, then we can actually calculate the genotype frequencies using the Hardy-Weinberg equation, which is shown here, p squared plus 2pq plus q squared equals to 1. The way that this is kind of derived, you can probably see in the table here. So you've got your capital A, your lowercase a, your p, and your q, and you basically just multiply it together to get all of your values. So then your expected genotype frequencies, your p squared, essentially represents having 2 capital A's, 2pq will represent a capital A and a lowercase, and q squared is going to be your 2 lowercase. And your Hardy-Weinberg law, it's essentially used to predict your genotype frequencies and also estimate your carrier frequencies of autosomal recessive disorders. So I believe you guys would have covered this in one of your lectures, but if you guys have any questions of that, feel free to chuck it in the chat as well. So this is kind of a little bit of a summary as to what exactly is allele frequency versus genotype versus phenotype frequencies. So if we have a look here at the recessive or 2 recessive alleles, your 0.45 would be representing your allele frequency. And in order to figure out what the genotype is, you would do Q squared, which is 0.45 squared, so 0.2025, this would be your genotype frequency. Likewise, so would these be for the respective genotypes. If we were looking at phenotype frequency, you'd be adding these two values together because both of these will kind of show themselves as the same phenotype, if that makes sense. Yep, so previously we were talking about using the Hardy-Weinberg equilibrium equation, sorry. So now if it's not an equilibrium, then it can be assumed that there might be other factors that are influencing this. So what are these different factors? Well, first of all, mutation. If you've got a mutation in your gene, this is going to cause your Hardy-Weinberg equilibrium to be disturbed. Likewise, migration. If a large population migrates from somewhere to somewhere else, that's going to add new alleles to a gene pool, so that's going to change your frequencies as well. Positive, negative selection. This is basically saying if a particular gene has a significant advantage over another one, that's going to affect how many of those genes that will remain in your gene pool. Genetic drift or bottleneck, that's essentially saying, so let's say in a particular population, if there's been some kind of disaster, killing of majority of people and leaving only a small proportion behind, this particular group, that's not going to be representative of what the original population, and that's going to affect your Hardy-Weinberg equilibrium as well. And non-random breeding, so if you're inbreeding, so within the family versus outbreeding, well, that's going to either increase your homozygosity or increase your heterozygosity. So, yep, these are just a couple of factors that will influence it. So a couple of questions. First of all, question 1, what is true of populations that are in Hardy-Weinberg equilibrium? So 1, natural selection is occurring. B, the populations must be very small. C, mating is random. And D, organisms are constantly migrating. Yep, C, awesome. So I think this is pretty much, it's pretty self-explanatory, but I'll just quickly go through it. So population must be small. No, your population has to be large. The organisms are constantly migrating. No, that's not true. That's going to affect it. And as for A, it's kind of true, but it's not too relevant to your Hardy-Weinberg equilibrium. So yeah, C is correct. Question 2, so a population of sheep is in Hardy-Weinberg equilibrium. The allele for white wool, W, has an allele frequency of 0.2, and the allele for black wool, W, has an allele frequency of 0.8. What is the percentage of heterozygous individuals in the population? Okay, so if we've gotten one answer of B, that is correct. So B, did you guys want me to run through how B is the correct answer, or are you guys good with that? Does someone want to say something in the chat? Not too sure. Okay. Well, okay, I'll quickly run through it just in case. So essentially the way that you are, actually, I've got something, maybe just run through it. Okay, no worries. So, actually, it might be better if I write it out. Let me see if I can change the screen sharing. Just one moment. Okay, sweet. So hopefully this will work. If I can get the values up. Okay, no worries. So we're essentially looking at the allele for the white wool and also the black, the frequencies of those two. So we know that the white wool is dominant, so let's call that P, and we know that the frequency is 0.2. And the allele for the black wool, so that's recessive, so we'll call that Q, and that would be 0.8. Yeah, so in order to work out the percentage of heterozygous individuals, you essentially are looking for 2 PQ, in which would just be 2 times whatever proportion it is for P, so that would be 0.2, and then whatever proportion it is for Q, and that's 0.8. When you multiply those together, so this is just maths, essentially, you get 0.32. So hopefully that makes a bit of sense. And now I have to switch back. Okay, sweet, so that should be all right. Okay, question 3. In corn, purple kernels are dominant to yellow. A random sample of 100 kernels is taken from a population in Hardy-Weinberg equilibrium. It is found that 9 kernels are yellow and 91 kernels are purple. So what is the frequency of the yellow allele in this population? And I'll give you guys a minute to work that out. Okay, so I've got a few B's, also a few C's. Correct answer is actually C. So this one is quite a difficult question. It does tend to throw people off. So the way that you would work this out, just saving time so we don't have to go back and forth from iPad to the screen. So essentially, if we're thinking about yellow, right? So this is going to be your genotype. So the genotype for this would be Q squared because we're thinking of recessive. So Q squared would equal to 0.09. And when we're just looking at the allele frequency, we're looking for Q. So then you would actually do the square root of 0.09, which would give you 0.3. So hopefully that makes a bit of sense. And we'll move on. So human karyotypes. So what essentially is a karyotype? It's essentially an image of all of the chromosomes in a human body. So any kind of tissue can be used to basically take a photo and get a karyotype up. The way that you would describe it is the number of chromosomes, your sex chromosomes, and then any kind of abnormality that you kind of observe. So an example of this would be 47, 2 X's plus 21. This would be a female with Down syndrome. So as you probably know from your previous lectures, so humans, we've got 46 chromosomes, 23 pairs. Amongst those, 22 are autosomes and 1 pair are your sex chromosomes. So for females, it's 2 X's. For males, it's an X and a Y. And talking a bit about your chromosomes and their structures. So from your kind of PSP lectures with Isabel, you should know, so your DNA fibers, you essentially wrap them around your histones, and those will produce nucleosomes. Nucleosomes, afterwards, they can condense to form chromatin, and chromatin can come together to form your sister chromatids, and that together forms your centromere. Oh, sorry, no, forms your chromosome, which has a centromere in the middle. So your chromosome structure, you've got 2 arms. You've got your P arm and your Q arm. So your P arm basically is your short one, your Q is your long one. The P, it stands for petite, and if you did French in high school or know French elsewhere, basically just means short. So that's a good way of remembering it. And each arm, it's capped by a telomere as well. And then about G banding. So G banding is just a way for us to tell what chromosome, basically label what chromosomes they are. So every single G banding, it's going to give you a unique kind of pattern. And from those patterns, we will be able to identify what chromosome it is. But yeah, you don't have to know too much about it. It's just something that they use to figure out what chromosome they are. So classifying the chromosomes, the way that the lecturers did it, I think they only mentioned 3. I've included 4 just for the sake of completion. You don't really have to know about telocentric ones, but yeah, it's there just for the sake of completion, like I said. So going through this, we've got our metacentric chromosomes. That's just with your centromere in the middle. So think M for middle, M for metacentric, I guess. Submetacentric, that's between the middle and the end. Acrocentric, it's the ones with the little satellites on the top. And telocentric is just your centromere at the very end. So this is a summary table just of all the nomenclature. A lot of it is self-explanatory. So D-L for deletion, I-N-S for insertion. I'll leave you guys to kind of have a look at it in your own time, but it's more something that you can refer back to if you ever do get confused about all the shorthand notations. Okay, now going on to the chromosome abnormalities, this is something that you do have to know, and it will be covered in your shoots, hopefully, as well. So chromosomal abnormalities, we can split them up into 2. So first of all, your numerical abnormalities and structural we'll talk about in the next 5 minutes. So numerical abnormalities, you've got your aneuploidy and your polyploidy. So aneuploidy just means you're missing a chromosome or you have an extra one. So we've got monosomy, that's loss of one chromosome. Trisomy, you've got 3 in total, so you've gained 1. And tetrasomy, you've got 4 chromosomes, so you've gained 2. So it usually results from non-disjunction at anaphase. Non-disjunction just means it hasn't split apart properly. And if aneuploidy, if it occurs during mitosis, you've got something called mosaicism. So what mosaicism means, it's essentially if you have cell lines with multiple karyotypes. So as you can see in this image here, you started off with 2 chromosomes, and then when they split, they didn't split properly. So now you've got 3 here and 1 here. And once your cells undergo mitosis, you'll end up with 2 different cell lines, each of them having different number of chromosomes inside, and that can be quite detrimental to individuals as well. So your second type is your polyploidy. That's just a multiple of your original number. So if we think we usually have 2 pairs of 23 chromosomes, here you've got 3 of those 23, so it's triploidy, you've got 69. In this case, it's going to be lethal. Normally you can have polyploidy cells for certain cells in your body. So example of here that I've chucked in is megakaryocytes. That's essentially the bone marrow cells in your body. So those can have polyploidy, or those can be polyploidy, but if you've got 69 chromosomes in your, germ cells, that's going to be a problem, and the fetus probably won't end up surviving until birth. So that was all your numerical abnormalities. This is your structural. So structural, we can divide it into your balanced rearrangement and your unbalanced rearrangements. So if it's balanced, it's usually all right because you haven't gained or lost any kind of chromosomal material. Versus unbalanced, this is going to prove quite detrimental because you're either missing certain genetic information or you've got too much of it. And yep, as it's written here, you're going to have severe clinical effects. So examples of this, all of your unbalanced rearrangements, this is going to include deletions, duplications, and insertions. So you're either going to be missing or you're going to have gained extra chromosomal material. Sorry. Yep, and balanced rearrangements, inversion, so that's just swapping it. Where's an example? There we go. Inversion, so we've just swapped the locations, but all the chromosomal material is still there. And translocation, it's basically you're changing locations, but once again, all the material is still there, and we'll talk about that in a little bit more detail here. So types of translocations, we can break it up into 3. You've got your reciprocal ones, where you basically just swap it. You've got your Robertsonian translocations, where the tips break off and then the 2 long arms fuse together. You can think about it in the sense of like Robertsonian, so they're sewn together. And insertional translocation, where you've got one part and you basically insert it into a different chromosome. So all of the information, once again, is still there. However, it's just in a different location. So DNA mutations, once again, was covered in a previous PSP kind of, I wouldn't say lecture, PSP session. But yeah, this is just a bit of a summary if you guys have kind of forgotten. So point mutations, they can be silent, in which there's like no change. Missense, in which amino acid has been changed, so your protein function is going to be changed. Nonsense is when you've got a stop codon, so no proteins produced. And frame shift, probably the worst of all. If you've got a deletion or an insertion of a base, then everything's going to be shifted backwards or forwards, and then it's going to produce something either very weird or no protein at all. And all of this, it can basically lead to a loss of function, gain of function, or something called conditional mutations, where in a certain environment, you're going to get a particular mutation. And if you're not in that environment, it actually might not show itself. There is a link, I think I've attached it to the speaker notes of the slides. So if you guys access that through the Google Drive, there is a YouTube video on that if you guys want more information, but it's not too high yield, so up to you guys. So now for the genetic disorders, these you will have to know. So all of your autosomal ones are here. So you've got your Edwards syndrome, trisomy 18, Patel, trisomy 13, Down syndrome, trisomy 21, and Kreutzfeldt, a deletion of your, a proportion of your chromosome 5. So Kreutzfeldt, that's quite an interesting condition. It's basically where you've got a proportion of your chromosome 5 being deleted. If you look at this, it's basically P being the P arm, 5 chromosome 5, and the minus meaning deletion. And what happens is this is a syndrome that affects the larynx of this child. So all the infants that have this, they will typically have a high-pitched cry like a cat. So if you ever see like the words high-pitched cry or like sounds like a cat, that's kind of your buzzwords. So you should immediately be thinking Kritisha. As for the other ones, a way to remember it, I guess, Down syndrome, it's had a lot of media coverage. So in a sense, I think that's how I just kind of knew trisomy 21, it's been thrown around. This one's a very stupid one. So Edward syndrome, I was like, okay, Edward from Twilight, he was actually 17, but I was like, oh, close enough to 18. So that's like trisomy 18. And for Patel, trisomy 13, that's just whatever's left over. And these are your sex-linked ones. So you've got your Klinefelter syndrome and your Turner syndrome. So Klinefelter's, you've got 2 X and a Y. Turner's, you've just got an X. So quick question, what is Patel syndrome? Yep, B, awesome. And these are just the rest. And question 2, which of the following will have the least severe clinical effects? B? Yeah, cool. And these are just the rest, all being unbalanced. Okay, and this is basically just a bit about a Punnett Square. I'm quite sure majority of high schools should have covered this. Yes, I probably won't spend much time on this, but if you are confused, do check out this link. It basically takes you to Khan Academy, and it's also covered in your active learning as well. So you can have a look at that, but otherwise, essentially just put your genotype or the alleles of parent 1 here, parent 2 here, and then you just put them together, and then you can work out the probabilities. So I'll let you guys have a look at that in your own time. Pedigree, so this is what you guys either have covered already or you guys will be talking about it a bit more in your active learning next week. So autosomal dominant, you will essentially have no skip generations. Everyone who is affected has to have at least one affected parent. Autosomal recessive, people can be carriers, so it can skip generations. X-linked dominant, females will most commonly be affected, and an affected father will always produce affected daughters. And X-linked recessive, well, males are going to be more commonly affected because they only have one X chromosome, and your affected mother will always give that affected X chromosome to the son, so the son will always be affected. You've got your Y-linked, which is characteristic of all the males, so you're not going to see any women with Y-linked issues. And mitochondrial ones, they're always going to be affected from your mother. X activation, you don't really have to know too much about this. It's more saying that for females, you might have less severe kind of presentation because one of your X chromosomes can be kind of inactivated. But yeah, you don't have to really know about that. So the 2 conditions that are covered in your active learning. So first of all, cystic fibrosis, it's also going to be reiterated in your ICL, so don't stress too much about it if you don't understand it, but it's basically you've got a mutation in your CFTR gene, and CFTR, it normally allows your ions and water to flow in and out of cells. So what you get is, if it's not working properly, you're not going to get enough water in your mucus, so then you end up with a very thick layer of mucus in your lungs. So for people with cystic fibrosis, you'll often hear them like coughing, and they'll be producing a lot of very thick phlegm. And the second one is familial hypercholesteremia. So familial runs in the family, hyper means too much, cholesteremia, that's just cholesterol essentially. So autosomal dominant, it's a mutation in your LDLs, and because if you have too much cholesterol building up in your blood vessels, you're going to end up with atherosclerosis and also increased risks of CVD. So quick question, and I think it's the last one as well. What kind of inheritance does this trait follow? Yep, perfect. So autosomal recessive, sounds good. The reasoning behind this is just because it skips a generation and there's an equal number of males and females affected. And you guys will get a few more examples of these in your active learning as well. But yeah, if you go to the previous slide, this one and also this one, you can basically have a look at those and you should be able to work out what kind of pedigrees or what kind of conditions they are. So that brings us to the end of genetics. Thank you guys for tuning in. If you have any kind of questions, I'll be sticking around for the next 2-3 minutes. So either send a message in the chat, or if you guys have any other questions, feel free to message me as well. But otherwise, thank you guys and see you guys next week.